And thank you for joining us today for the Human Cell Atlas Biological Network seminar series. Today we're featuring reproduction. A couple reminders before we get started. Please, if you're not a member of the HCA, please visit our webpage and join our membership. And as you do that, you can select any of the 18 biological networks to become a member of. You can select more than one, and that will allow you to participate in the Atlas building and receive all of our communications about it. If you are planning to publish and would like to have your publication to be included as an HCA publication, please visit the publication link and submit a short form, and it will help your paper gain more visibility. A couple announcements of the upcoming HCA events. The HCA Town Hall is on June 5th. It's a virtual event. We will announce the registration shortly. Please join us to hear the updates on the HCA progress over the past several months. The HCA General Meeting this year is in September in Milan. The registration is now open. Please visit the events link on our webpage to register. HCA Asia meeting is in Hong Kong, December 3rd and 4th. And HCA General Meeting next year, 2025, will be in Singapore, and we'll announce the dates shortly. If you would like, to sponsor the meeting or exhibit there, please email us and we'll connect you. Today we have a packed seminar agenda. We have 4 talks. So our presenters today are Vento-Tormo, Ariella Shikanov, Cecilia Lindskog, and Santhilal Subhash. And I would like to introduce our first speaker, Rosa. So Rosa leads a research team at the Wellcome Sanger Institute. She's also co-chair of the HCA Reproduction Bionetwork. Rosa's research is focused on the adaptation of immune cells and tissues and their function in steady state and inflammation. Her team has a special interest in modeling cellular interactions and uses single-cell spatial transcriptomics and functional assays to reconstruct the signaling mechanisms underpinning cellular behavior in healthy and diseased tissues. And Rosa is a co-chair of the Reproduction Bionetwork at the HCA. Rosa, welcome.
Thank you very much for the nice introduction, and hopefully you can see my screen.
Yes, perfect. Okay. Go ahead.
Fantastic. So today I will be talking about our effort to map the human uterus in time and space. And why are we interested in the reproductive system? And the main thing is, so far has been really a black box on how it develops, how it regenerates, and how it functions. And this is due to main causes. The first one is due to the intrinsic difficulty of accessing human tissues, and particularly for the uterus and the pregnant uterus, that's, as you can imagine, quite complex. The second reason is because reproductive biology is really under-researched. And just few numbers, there are 4.5 more papers in non-reproductive organs than in reproductive organs. And also there is an evident funding gap in women's health. And that really has a repercussion on reproductive biology. Third, there are quite a lot of differences between the reproductive tissues in humans and mouse, and that includes differences in the physiology and hormonal and function of the tissues. And fourth, there is still a lack of complex in vitro models. There are some in vitro models starting to appear, like epithelial organoids, but the complexity is not recapitulated on them. And all of this really has an impact on women's health because reproductive conditions are widespread. Just to give some numbers, differences in sex development affect 2% of the populations. Congenital uterine abnormalities, about 7%, and endometriosis, 10%, and there are others. And all of this really causes a significant morbidity in women as well as infertility. So I will focus a bit on endometriosis, which is a disorder that I discuss in my talk. So endometriosis appears in 1 in 10 of those born with a uterus. And what causes this is the cells resembling the endometrium are found outside the uterus. And really, this causes debilitating chronic and cyclical pain. Not only the tissues where you find these endometrial-like cells are affected in endometriosis, but also the endometrium itself in the uterus, it's also affected. And this really causes infertility. In these women, we can see infertility, and it's quite frequent, and the menstrual pain. And despite the impact, it still takes us a mean of 8 years to be diagnosed, at least in the UK, and there is no efficient treatment so far. So obviously there is an urgency to really study endometriosis and find biomarkers. So the way that my team have been about filling this knowledge gap in reproductive tissues and reproductive disorders have been the use of human cell atlasing to profile the reproductive organs across the lifespan in humans. And we have been mapping the ovaries here in 2022, also the endometrium, both on the non-pregnant endometrium and the pregnant one. And we have also been using in vitro models such as organoids to validate the hypothesis that we make from these atlases. And for today's presentation, I'm going to focus on our team's work in the adult non-pregnant endometrium, which will be a summary of these 2 manuscripts. So the human endometrium is this mucosa layer of the uterus, and is comprised by an epithelial layer with glands that is surrounded by fibroblasts and immune cells, and also vascularized tissue. And what is very cool about this tissue is that despite its complexity, it regenerates cyclically in response to ovarian hormones, which is the estrogen and progesterone. And each regeneration is a perfect regeneration of the epithelial tissue and also the surrounding the stroma and vascular tissue. It occurs without fibrosis, without scarring. And this happened really more than 400 times of cycles over a woman's lifetime. And as you can imagine, has to be very tightly regulated. So in 2021, what our team asked what are the main signals that drive the regeneration of the endometrium. And for this, what we did was to use single cell and spatial transcriptomics profiling human samples. So we focused mainly on the epithelial cells, which are these epithelial lumen and the glands. And here are the single cells from the epithelium in both the proliferative phase when the tissue regenerates and in the secretory phase where the tissue differentiates. And as you can see, we discovered new cell types, which here you can appreciate the different colors. And really was the spatial data what allowed us to give an identity to these cell types. So, for example, in the proliferative phase, we found that there are 2 populations of epithelial cells. The ones that are LGR5 negative and the ones that are LGR5 positive. And taking advantage of the spatial data, we could map this LGR5 positive in the lumen of the tissue and this LGR5 negative in the basal layer of the endometrium. So those are two candidate progenitors, and really by the convolving the bulk RNA-seq data from endometrial cancer, we could see that this LGR5 positive, which is located in the lumen, is associated with poor outcomes of cancer. So then we moved forward into the secretory phase. So we have the tissue regenerated and now it needs to differentiate. And again, taking advantage of the spatial transcriptomics, we could see how, again, there is a luminal population, again, LGR5 positive located in the lumen, and the ones in the glands that are LGR5 negative. And also we could see another population of this of epithelial cells that are the ciliated cells that also collocated together with the luminal LGR5 positive in the endometrium. So that was very interesting. And one of the reasons that was very interesting is that then we could look into the pathways that regulate this epithelial heterogeneity in the uterus. And for this, what we used was CellFundDV, which is a tool I developed during my postdoc in Tachman team to look for ligand receptors interactions. And what we could find is that the surrounding stroma creates two different niches in the endometrium. Close to the lumen, there is a signal increasing the Wnt signal, while surrounding the glands, the fibroblast signal in a way that there is an increased signal of notch. And this balance of the Wnt and notch is essential for the epithelial identity. And to demonstrate that, I'm not going to show you here, but you can check in the manuscript. What we did was to use endometrial organoids that really showed us how this disbalance in the tissue by supplying these small molecules to inhibit Wnt and notch, can we create only one or another epithelial subset. Good, so that was a very interesting finding. And after mapping this kind of basic distribution of epithelial cells in the tissue, what two members of my team asked, Luz Garcia Alonso and Magda Marekova, is how our atlases correspond to other atlases that have been produced at the same time and that are publicly there, and that maybe don't have the spatial data, so the annotation is not that clear. And even more interesting, can we learn more subsets by integrating new data, because then we will have more data. So to really take into this gigantic effort, what the problem was that when we integrated a different data set, it was difficult because there were a lot of batch effects. So what we did is that Luz Garcia and Magda Marekova designed an experiment where we did create our data set that have anchors from the different data sets. So we included a bit of samples that were from proliferative states, another from the secretory, and others treated with hormones, and others treated without hormones. And in this way, we have an anchor that allows us to include all the different data sets and really overcome the batch effect that we will have by integrating the data sets. And with this, we created this data set where you can appreciate the different colors, really showing the multiple identities and new identities of epithelial and mesenchymal cells that we found in our atlas. Also, to validate the data, Luz and Magda generated a nuclei data, and this was from a lot of patients, and really this didn't have any batch effect, and we validated the new populations in these alternative data sets. So with all of these, we created this atlas where we find, and also by, I forgot, of course, by looking at the spatial data and where each of these population is located in the space and time, we could create this new map where there are a lot of new identities and where are they located of epithelial cells and fibroblasts. And one of the things that you can see is how variable these populations are across the different stages of the menstrual cycle, really showing how when you study disease, you really need to look into the stage of the cycle, in the same stage of the cycle for both health and disease. And with this map for this presentation, I'm just going to show you a few interesting results, and there are more in the bioRxiv paper. Basically, for example, what we could find is to define the epithelial populations in the proliferative, so that when the tissue is regenerating. And what we found by, again, looking at the spatial data is how we have in the basal layer, what we defined before as this LGR5 negative, now we have 1, 2, 3, 4 populations. And really, the one that is in the basal layer and that has more progenitor-like features is located just here and expresses CDH2 positive. They are CDH2 positive, while the ones in the glands, in the regenerative glands, express other markers. And again, this is important because now we can start looking into the signaling that these different progenitors receive. So, for example, collocating these SOX9 basalis, CDH2 positive, we have a specific population of fibroblasts that are called C7 and that we discovered in the 2021 study. And we could see how they are signaling this kind of stem cell niche through the CXCR4 axis, also through Wind axis and FGF7. Good. So with all of this, we have defined this atlas and really focus on this proliferative stage and how there are different niches and different progenitor niches and how these fibroblasts compartmentalize the tissue and give the specific signal to the progenitors. So then we also look at the immune populations in the endometrium, and we focused on the macrophages, which are mostly enriched again in the proliferative phase, the phase of regeneration. And you need to think about this regenerative tissue when there is menstruation, there is all this inflammation and wound healing that is really necessary for the regeneration of the tissue. And during this phase, what we found in our data is that there are specific ligand receptors that enable these macrophages to come and to the tissue, and how these macrophages increase the wound healing response that is important for the regeneration of the tissue. And at the same time, they start decreasing inflammation. And that's also necessary because we need to remember that this regeneration occurs without scarring, so there is no fibrosis in the tissue. The other thing that we found is that macrophages also interact a lot with the perivascular cells and the endothelial cells with the vascular niche, and secrete angiogenic molecules that are likely to be essential for this vascularization of the tissue that really needs to happen, because in the future, this vascularization is really important for the embryo implantation and development. So with this, we defined a role for macrophages in the regeneration of the tissue, as well as together with the different epithelial progenitors. And really, the final part was in the tissue. We also, in the map, we also included from our end and from others data from endometrium, not only contours, but also from endometriosis patients, which I told you that also have effect on infertility, for example. And we didn't find a new population in the endometrium from endometriosis patients, but what we found is that there was an increase of macrophages in the endometriosis cases and a decrease in the fibroblasts that are responding to hormones. And that was quite interesting. And what we could find is that when we cross it with GWAS data, we also found and looked for SNPs and we also found this enrichment on the fibroblasts and macrophages, showing that those are the likely populations affected in endometriosis. Really showing that in the endometrium from endometriosis patients, there could be an inflammatory response altered, as well as an altered response to hormones. And indeed, what we found that these fibroblasts, the genes affected in this fibroblast that are responding to hormones, really are genes related to the signal that they will give to the epithelium in a way that in this patient, probably the epithelium differentiation doesn't go as well due to the wrong signal of the fibroblast. And with all of this, we conclude our work in which we have focused on the fundamental biology, looking at new cell populations, the regeneration of the tissue, the role of macrophage and epithelial stromal signaling. And we have found these altered Wnt signaling and inflammatory pathways in endometriosis, which we are hopeful that it will help on diagnosis and design new treatments. And moving forward, we are excited about a comprehensive profiling of endometrium and lesions from multiple patients, as well as using these atlases to develop advanced in vitro models that allow us to validate all these findings. And with this, I just want to thank my lab, and in particular, Luz Garcia Alonso, Magda Marekova, and Luis Francois, who have lead the different projects together with collaborators in Oxford and Cambridge University, as well as Sanger. Thank you.
Thank you so much, Rosa. We have a couple questions and a couple minutes to answer them. One question is from Alyssa. How do you obtain your endometrial tissue samples?
Yeah, there were these biopsies from coil biopsies. So yeah, just biopsies.
Great. And another question is from Gamal Akabani.
Well, I will say it's not only, sorry, it's not only biopsies, but from the 2021, which we also integrated, we also have uterus from donors who die from non-gynecological pregnancies, and that's why how we also can map out the layer of the layer of the basal layer, which doesn't which stays and which you don't get through the biopsies.
Mm-hmm.
So it will be both biopsies plus plus the full uterus from these donors.
Thank you. The next question is, we always assume that homeostasis is caused by estrogen and progesterone balance. Are there other factors that after the homeostasis of the endometrium cycle that alter the homeostasis of the endometrium cycle, such as cellular population imbalances?
Well, I'm sure, and also there are signaling that interfere with the hormone themselves. So for sure, other signaling and other inflammation would also affect this. So yes, both population imbalance and as well as other signalings that feed into the hormones will cause may cause some pregnancy complications. And for this, with the population disbalance could be immune cells that are infiltrating and that can cause inflammation, for example. That could be one thing that could be affected.
Great. Thank you so much, Rosa. If there are any additional questions to Rosa, please type them in Q&A section. And maybe, Rosa, you have a couple more minutes to respond. Meanwhile, I would like to welcome our next speaker, Ariella Shikanov. She is also co-chair of the HCA Reproduction Bionetwork. She is an associate professor of obstetrics and gynecology and biomedical engineering at the University of Michigan. Ariella's group is focused on solving the clinical problem of premature ovarian insufficiency, which is a common outcome of anti-cancer treatments in young girls and women. Ariella's lab is also interested in molecular mechanisms involved in early stage development of ovarian follicles, development of novel 3D culture systems, and immune-isolating hydrogels for ovarian follicles. Ariella, welcome. Thank you for joining us today.
Hi, hello. Thank you so much for such a kind introduction, Ellen. I appreciate it. So today I will be telling you about our most recent publication, mapping human ovary. So this was funded by the HCA, CZI. It's a collaborative project between multiple labs. So Sue Hamoud, and then June Lee, and myself, and then the two students who really spend a lot of time working on this project are Andrea Jones. She just defended her thesis a few months ago, and Ford Hannum in Dr. June Lee's lab. So I'm actually very grateful for Rosa to mention. I don't have a slide telling you all about how difficult it is to get human ovarian tissue from young women, especially reproductive age women. Nobody wants to give up their ovaries. And similar to Rosa, we also in the United States experience very low numbers of publications and labs who work in this area, and clearly funding is also quite tight. So we are very grateful and fortunate to have this funding to really pursue questions related to female reproductive biology, especially related to ovarian tissue. So this beautiful image was taken by Andrea using nanostring technology. So we used spatial transcriptomics to analyze ovarian tissue from 2 donors, and also single cell RNA sequencing from 3 additional donors. So what are the questions that we were interested to ask? So if you look at the ovary, and there have been a few papers published doing single cell RNA sequencing on the ovaries, but then ovary really have this very unique morphology. So ovaries have ovarian follicles. So this is the functional unit of the ovary. They can be found at different stages. So majority of the ovarian follicles are at the primordial stages, and they're located in the cortex. There are also other early stage follicles, such as primary, transitioning primary, and also secondary follicles. So if you just look at the cross-section, morphological cross-section of the ovary, you can see all these follicles very easily. But then when you go and do single cell RNA-seq, you kind of lose this spatial location of these, the information about where these cells are coming from. There's also secondary follicles. Vasculature is also very rich in the ovary, mostly in the medulla part, which is the inner part of the ovary. And then ovarian surface epithelium is also very important, especially for people who study ovarian cancer, and cysts or corpus lutea. This is what's left over from ovulating follicles. So kind of using spatial transcriptomics, we were able to go and target all these areas by collecting this guided selection of regions of interest. I'm going to call them ROIs from now on. And again, so you get what you get. So we had 2 donors, and we were super lucky because those donors had large antral follicles. So when you look at the large antral follicles, we were able to go and collect RNAs from the oocyte, and then we were able to target the first layer of cumulus granulosa cells, and then the second layer of cumulus granulosa cells. And you can see all these numbers here. So the numbers 11, 12, 13, they correlate with the ROIs that I will give you the information in the next slide. And we were also able to target mural granulosa cells. So these are cells further away from the oocyte, and also theca layer as well. We also were really interested in looking at the primordial follicles. So again, when you do single cell, so oocytes are large. So starting at the secondary stage of the follicles, oocytes reach about 30 and up micron in diameter. So it's really, they cannot be sequenced individually. However, here using spatial transcriptomics, we were able to label oocytes in primordial follicles using antibodies. Here we use dazzle antibodies and then collect all the transcriptomics from those dazzle positive regions and then compare it to dazzle negative one. We also were able to look into other enriched regions such as CD68 to target macrophages, smooth muscle cell, and also progesterone-enriched regions. And because we could just kind of take a whole piece of the ovarian tissue, we were able to target this surface epithelium and also regions going inside the ovary. So kind of collecting these layers, sequential layers from surface epithelium inside the ovary to try and identify potential gradients of transcription. So here's the overview of the spatial transcriptomics. So as I mentioned, we had two donors, donor 1 and donor 2 on the right. The donor 1 was 18 years old young woman, and we had both ovaries arriving into our lab, and donor 2 was 28 years old. And here you see, so this is the cross section. We call them salami slice. So it's basically cross section across the whole ovary. And what I highlight, at least in this section, is this beautiful antral follicle, antral follicle, pre-ovulatory with oocyte cut into this section. So again, we're super lucky. And then you can clearly see the morphology, all these layers of cumulus granulosa cell, and then bural granulosa cell and theca layer. So by targeting and creating those ROIs, we could kind of really answer some of the questions related that can compare structure and function. In the second donor, we were very excited to see multiple clusters of primordial follicles. So here they're pointed with this red dye, so it's dazzle positive. So we were able to stain the whole section with dazzle antibody to be able to collect transcriptional data from these specific oocytes. So when you look at the analysis of the transcription, so this is donor 1 and donor 2, every dot correlates with ROI, right? So this is multiple, it's not single cell, it's multiple cells. And what was super exciting and fascinating for us that actually these regions correlated. So there was really nice agreement between the two donors. So in donor 1, we only had one large antral follicle, and you can see it here. So this is one dot, purple dot, and this is the oocyte. And then in donor 2, we had multiple oocytes from primordial follicles that were dazzle positive. So here, all the purple dots are correlating with the primordial follicles, so in donor 2. And then we also looked at the different locations in the ovary. So here you see ROI type in this table below. So we looked at the ovarian surface, cortex series, primary follicle rings, so everything around the follicle but the oocyte, secondary follicles, and then also some of the cysts. And then these are the orange dots. So here, this is the cortical series in this donor, and then some of the cortical series in this donor. Then we focused on the medulla parts. So medulla has, it's kind of more unidentified region. So all the blue dots correlate with the medulla section in donor 1 and then donor 2. We were excited to see the theca cells, so we could go with specifically and collect theca cells, and these are the yellow ROIs in donor 1 and donor 2, and you see how beautifully they separate from the rest, and then the granulosa cells are green in donor 1 and donor 2. So we got pretty nice agreement between the two, and then we went in and really started looking into more depths into these regions. So this is the antral follicle from donor 1. Again, you see the oocyte. So this is just the H&E staining from the next section. So this is like 5 micron away from this section, and this is the section that was submitted into the nanostring instrument. So again, each dotted line correlates with the ROI that was analyzed using nanostring. So each layer is correlating. So the oocyte, and then the outer layers of cumulus granulosa cells, and then you have some other granulosa cells that are not as close to the oocyte, and then further away mural granulosa cells. Here, the two regions, and then thicker cells are in yellow dotted line. So looking into this, so what are the genes are saying? So this is the zoom in, so you have 4 different layers, and it was fascinating for us to see that they really created this heat map that the gene expression is very different, right? So going from the oocyte out, you see overexpression of certain genes as you move away from the oocyte, which, and these genes correlate with meiosis, PKA signaling, steroidogenesis as expected, and also response to luteinization. Further away, kind of comparing the granulosa cells and thicker cell expression, what we see here, there was a very nice agreement between the 2 donors, similar to with the other ones. So granulosa cells are a lighter green, so anything but it's not cumulus. The cumulus granulosa cells are darker green, so darker green here and darker green here. The lighter green is just general granulosa cells, and then the thicker cells cluster very separate from the granulosa cells, and they're in yellow. Looking at the heat map for these 2, you can see again clear separation. So the thicker cells are this column, and then the granulosa cells is the green column from donor 1, and similar view you can get from the donor 2, again thicker and granulosa, and really very nice gene expression. Some of the genes are expected, some of the genes, so CYP17, CYP19 would be expected, but some of the genes were unexpected, and we're doing further validation to understand what's happening there. We were also very interested to look at the gradient, right? So we talk about, whenever we talk about ovary, we think about 2 major regions. So you have the cortex, which is follicle rich region in the ovary, and you have the medulla, which is more of the blood vessels, immune cells, larger follicles, or corpus luteum. So what we wanted to do is to look in depth, what is the differences between these layers. So what Andrea did is she took this region, small square, and then starting from the surface epithelium, she took these 30 micron deep sections going from the cortex, from the surface epithelium inside the ovary, collecting 11 ROIs. So the total width of it's 350 micron, and then this is 30 times 11 is 330. So what is the gene expression? And we were, again, I mean, we were surprised, but pleasantly surprised. It kind of confirmed our hypothesis that there is a clear gradient for gene expression as you move from surface epithelium. So this is layer 1. As you go deeper into medulla, you have almost inverse expression of some genes. So you have increase in star expression. This is a steroidogenic responsive pathway. MMP2, which is matrix metalloproteinase. There's a lot of extracellular matrix related genes also change as you move inwards from the surface epithelium inside. So this is kind of the summary. So ECM remodeling, hormone signaling, and lipid signaling are all changing as you move in. Another important, I guess, feature that we were able to test using this spatial transcriptomics was looking at the oocyte. So early stage human follicles are very difficult. I would even say impossible to sample. Well, first of all, because they're so rare. So many of the cells that are kind of going under the radar are the primordial oocytes. They're rare, they're sensitive whenever you do single cell transcriptomics. Also some of the granulosa cells. So you have to have a lot of antral follicles to really identify granulosa cells and pull them out. Same goes with the thicker cells. The majority of the cells that you collect with using single cell are the stroma cells. So using this nanostring technology, we were able to take a piece of the ovarian cortex. This was donor 2, because donor 2, she had a lot of primordial follicles, stain them with dazzle, and then let the instrument collect dazzle positive, applying the segmentation mask. So collect all the dazzle positive regions and then collect all the whatever is not labeled will dazzle, and then compare. So using this approach, we identified the canonical oocyte specific markers that have been reported before and have been known, such as ZP3, OSP, LHX8, but we also were surprised to see some novel markers that haven't been reported before, and right now we're looking into kind of try to see and understand what is the role of these markers as well, but also looking at the heat map. We were again very pleasantly surprised and kind of confirming and validating our hypothesis. So the oocyte transcription really, this is donor 1, and then you have on the other side donor 2. They really look completely different, separated from the rest of the genes. And then another feature that we wanted to use, and we were super excited and used based on such rich morphology of the ovary, is looking at across the medulla and identification of the signatures. So this is the salami slice, as I mentioned to you, and we just went in and sequenced all these regions across. So this is M1, M1 going this way, M2 going this way, and then you have the cortical regions as well. And again, each dot here is ROI, it's not single cell, but it really separated beautifully. So all the medulla series here, on the cortex series here, and then the dorsal negative, which is the regions right around the follicle, labeled here in black, also separating separately. Again, so we were looking for potential progenitors for theca cells, potentially suggesting that maybe we can find something there. And then all the genes expressed here differently in these regions are labeled. We also did single cell RNA sequencing, of course, because we had 3 donors, and this is actually, to be completely honest, this is how we started, and then we realized we're not getting anything novel, so then we did the spatial transcriptomics, and very similar to the previous publications, whenever you do single cell for the ovary, you end up with this huge cloud of stroma cells that harbors a lot of cells, but they don't really separate. Our findings with the single cell confirmed what people have known before, so this immune cells, a lot of immune cells, pericytes, which are important for the endothelial cells and also endothelial cells, kind of probing deeper into the immune cells. We were able to identify macrophages, mast cells, natural killer cells, and T cells, and they separate beautifully here. And also here in the expression map, you can see all these different cells. What was super exciting to us that the oocyte and stereogenic signatures from spatial provided us with some insight into identifying location within the projection from the single cell. So this is the 3 additional donors, so donor 3, donor 4, donor 5, and the single cell map for each one of them. But we were able to apply our markers, identifying the spatial to the single cell maps, and we could kind of with more confidence identify granulocyte cells and theca cells. So to summarize here, I think I'm right at the time. We were able to, so using spatial transcriptomics, we were able to generate signatures for rare cell populations such as oocytes, especially from the early stage follicles, theca cells, and granulocyte cells. We would gain insight into transcriptomic signatures of follicle-rich cortex, so where the follicles are, and cortex gradients to better understand the early follicle environment, which supports follicle genesis and gives rise to theca and granulocyte cells. And single-cell RNA from healthy donors identify 4 major clusters and for immune cells, and also confirmed minor clusters, which has already been reported before, but kind of validated our findings as well. And overall, I think we built a contributed to comprehensive atlas of the healthy ovary that we continue working on this as well. So kind of spoiler alert, a student in my lab is now performing single oocyte sequencing to try to better understand the different stages of the follicles. I do want to mention one word. So I'm forever grateful for the funding. Thank you so much for the Chan Zuckerberg Foundation, my wonderful, wonderful collaborators, and my hardworking lab, and also the families of the donors.
Ariella, thank you so much. Wonderful talk. There are 3 questions for you. I don't know if we'll have questions, time for all 3, but whatever we don't get to, you can answer in writing. So one is, which spatial method will you continue to use?
This was so expensive. So we use nanostring genomics. I think it was bought by Bruker now, so it's Bruker. And it's a slide, and it allows you to do this regions. $10,000 per slide. Ideally, I would really like to do single cell spatial transcriptomics. I think it's not available yet, but yes, I would like to switch to single cell transcriptomics.
Thank you so much. There are 2 more questions. Please go ahead and answer them in writing.
I will.
Thank you. And meanwhile, I would like to introduce our next speaker. It's Cecilia Lindskog. She's also co-chair of the HCA Reproduction Bionetwork. Cecilia is a research group leader and associate professor at the Department of Immunology, Genetics, and Pathology and at Cancer Precision Medicine Unit at Uppsala University in Sweden. Since 2014, she has been director of the tissue-based efforts of the Human Protein Atlas Project, leading the team that generates the mRNA and protein expression data on human tissues. Her research group uses a combination of antibody-based imaging, transcriptomics analysis, and machine learning to generate high-resolution spatial maps of the human body at the single-cell type level. Welcome.
Thank you very much. So I'll talk about our ongoing efforts in my research group that are also highly connected to the Human Protein Atlas project. So in the Human Protein Atlas in general, we want to map all the building blocks of human tissues and cells and organelles, and we do that using a combination of transcriptomics and antibody-based proteomics. And all this data is done publicly available on the Protein Atlas website. And we have been working on this for more than 2 decades now. It started in 2003, and we have antibodies targeting most of the human proteins. The data in the database is divided into 12 different sections, each focusing on particular aspects of the human proteome, and you can then zoom in on different functional characteristics depending on the interest. And today I will mostly talk about work related to the tissue and single cell type sections, and in particular, the work done on reproductive organs. The tissue was the first part of the Human Protein Atlas, and we published our flagship paper in 2015, where we mostly focused on immunohistochemistry on tissue microarrays and then also bulk transcriptomics analysis. And that allowed us to divide all the genes into categories based on tissue enrichment, where we could then confirm these expression patterns based on antibody-based proteomics. And that covers the main cell types of all the main organs. And the images are all available on the website that then target most of the human proteins in the main tissues and cell types and organelles. And we'll look at one protein at a time. Then we've had a couple of follow-up projects on reproductive organs where we want to increase the resolution of the tissue-based data. So we looked at testes and ovary, where we either looked at the existing images in the Protein Atlas to add more detailed data to the existing images, but we also added novel samples, for example, from ovary, to target the follicles. As you already heard, they're really tricky to find in the tissue samples, and not all the images we had actually contain follicles. So to confidently map proteins that are expressed in the follicle cells, we had to add novel samples. And we also had a project where we recently added samples also from prepubertal women, so we could compare the expression between prepubertal and adult. And here the focus was to see if we could identify some proteins in these samples that had not been described before, that we could then also validate with other methods to assign a presumed function of those proteins that were not previously characterized. In an ongoing project on fallopian tube, we have, based on bulk RNA sequencing data, looked at genes that are elevated in fallopian tube compared to other tissues in the human body, and found that most of these are associated with cilia function. And we then did an in-depth study to study these with immunohistochemistry to define the localization in the ciliated cells to different compartments of the ciliated cells. And out of the 130 proteins that we analyzed, as many as 34 were not described before. And these were then also validated with single-cell RNA sequencing data and also in situ mass spectrometry. Here's some examples of the proteins that we analyzed, where you can see that we were able to confidently map the expression of those proteins to ciliated cells and also different compartments. And interestingly, when we compare the expression between ciliated cells in fallopian tube with ciliated cells in other organs that also have motile cilia, we could see that most of the proteins that we investigated were expressed in all motile cilia, so not only fallopian tube, but there were actually some proteins that were tissue specific, and we saw that choroid plexus was the tissue that was most different compared to the other organs with motile cilia. But then with the introduction of single-cell RNA sequencing and all the efforts in the Human Cell Atlas and also other consortia worldwide, we realized that we can go beyond the bulk transcriptomics data to look at enrichment also in single cell types in comparison to other cells in the human body. So we created a body-wide overview where we looked at enrichment in specific cell types. And that then allowed us to define genes that were enriched in, for example, ciliated cells and different compartments of other reproductive organs as well. But to take this to the next level and not only look at one protein at a time with regular immunohistochemistry, we have recently started to work more and more with multiplex profiling using antibody-based imaging, where we can then look much more in detail, because this method allows us to look at cell states and subsets that are not previously distinguishable by the human eye. We can also look at overlap and interactions, and it allows us to understand more about the cellular function. And another strength is also that this is more quantitative. So it allows us to do automated image analysis, which then has much more potential for translation and research. Move on to the clinical practice. So in an ongoing large-scale work where we're working on human testes, we have integrated single-cell RNA sequencing data from 2 different data sets, and then taken advantage of publicly available protein data with immunohistochemistry from the Human Protein Atlas, where we had almost 4,000 proteins with immunohistochemistry data. And based on that, we then built multiplex antibody panels that were targeting different subsets of cells in the testes. And then used these antibody panels, where we had a fixed panel of 5 antibodies each time, depending on the protein localization. So we selected the candidates to stain based on the existing immunohistochemistry. And then we added the candidate protein to this 5-plex panel, one at a time. So we could compare the expression of this protein with other 5 panel markers, and then analyze that based on automated image analysis. We used single-cell RNA sequencing data from 2 publicly available data sets, and then performed further subclustering, where we could identify 5 different subclusters of spermatogonia, 4 different spermatocytes, and 5 different spermatids. And then we used these clusters that we annotated based on well-known markers from literature, and defined antibody panels that matched with each of these different clusters for spermatogonia, spermatocytes, and spermatids, where we tried to, as much as possible, optimize antibodies that were non-overlapping, so that we could outline each of these subsets of cells And some of these would be very hard to distinguish by the human eye if we stain just one of them at a time, even for an expert. So this really allows us to pinpoint the spatial localization more in detail. When we then came to selecting the candidates to stain, we based this on the almost 4,000 protein candidates that we had available. When we clustered the stainings of those with a gross annotation that we had in spermatogonia, spermatocytes, and early and late spermatids, we could see that there were some that appeared rather specific to one or a few of those cell types that we wanted to investigate further. So we selected candidates that were as specific as possible based on immunohistochemistry data, and then from all the main categories, and came up to almost 500 candidate proteins that we wanted to stain. But as you see, some of them overlap between the panels that we tested. So there was 69 that we stained with both the spermatogonia and spermatocyte panel. And this was to really see when the expression started and stopped for these proteins. And we already knew that there were challenges in the immunohistochemistry annotation. So not until we stained this with the multiplex analysis, we were really sure in the exact location of these proteins. And here are just 3 examples, one of each panel. And based on automated image analysis, we were then able to get quantitative levels. So that's what you see as violin plots at the bottom, with the expression levels in each of the cell states that we had defined. And here we could see 3 specific examples that were then specifically localized mostly to one of the cell states. But since we stained quite many proteins, we were also able to group these proteins into expression categories. So here I'm showing examples of how it looked like for the spermatogonia proteins, where we stained 228 different proteins. And then we could see that we could identify 5 main expression clusters based on the automated image analysis data of the protein staining. And you see that these proteins were specifically localized to different combinations of these different cell states. And then it's interesting to see what type of proteins we find in each of these expression clusters. So we performed a functional enrichment analysis based on Go terms, and could then also see that based on the clusters and the expression in specific cell states, we also found Go terms that were enriched that mapped with what we expect in terms of transcriptional activity of these different cell states. But mostly interesting was that we, within each cluster, found quite many proteins that had not been characterized before. So this then gives a clue what type of function these proteins could have, and a starting point for further analysis, how these proteins are then important for male reproduction. But one could then ask what's the reason for doing all of this at the protein level if the single cell RNA sequencing data already exists? And one of the theories we had was that there could be a shift in the protein expression based on the transcription, because there are a couple of such proteins that are well known from literature. So for all of the proteins that we analyzed, we checked if there was a correlation between mRNA and protein levels according to the cell state specific expression, or if there was a mismatch. And actually almost 1/3 of the proteins that we investigated had some sort of mismatch in expression, and we found quite many where there was a delay. So the mRNA levels were found earlier, and the protein levels were then found at an later cell state. So the mRNA is stored and not translated until later in the differentiation process. And it should be noted that there were actually some proteins that are well known and described in literature as cell state specific proteins based on mRNA, where we could see that the protein was a mismatch. So this could be really important for further understanding the function of those proteins. In addition to testes and the male reproductive tissues, we're also working on some other projects related to the female reproductive system. We have some ongoing projects where we want to localize proteins in different cell types or subcellular structures of these different tissues, but also to see what are the expression dynamics through the lifespan of women. So we're looking at samples from different age and menstrual cycle. And I will just briefly mention an ongoing work by my master student, Filippa Bertilsson, where we have now developed a multiplex panel to outline the different subcellular structures of ciliated cells. So this is a follow-up study to the previous work in fallopian tube that I described previously, where we want to pinpoint this more in detail to define the exact localization in ciliated cells. So we stain 5 different structures of the ciliated cells with different markers and then add the candidate protein that we want to investigate to that in the same manner as the testis project. And we can then use automated image analysis to mark regions of interest based on these 5 different markers and measure the mean pixel value for each of the candidate proteins in these different regions. We're working on analyzing this data, but so far we stained almost 140 proteins, and in a similar manner as testes, we can then cluster the data into different expression categories where we look at the function. And here's just one example of a protein that was mainly expressed in transition zone and tip. And this protein is previously known to be expressed in cilia movement, but there is no literature where in the cilia it is expressed and exactly what type of function it has in motility. So as a summary, we're using integration of transcriptomics and antibody-based proteomics, and that allows us to explore the single cell state specific expression patterns in human reproductive tissues. And we believe that by understanding the spatial distribution of proteins, we will understand more about the biological and functional context of different proteins. And this then allows us to not only study the cellular tissue and cellular level, but also the subcellular level of the localization, which provides even more information about the function and context. And finally, I want to highlight that we think that the expression dynamics between mRNA and protein levels, especially in tissues where there is a gradient in expression or a differentiation process ongoing, it's important to further also look at the protein levels, and we should give this more attention and continue to look at this also for other tissues, I think, in the future. So with that, I would like to thank my team and also some previous members and collaborators and all of the Human Protein Atlas projects. Thank you very much.
Thank you so much, Cecilia. Wonderful talk. If there are any questions for Cecilia, please type them in the Q&A section. And Cecilia, continue monitoring it. Right now, there are no questions for you, but hopefully there will be in a little bit. Thank you. With that, I would like to introduce our next speaker, Santhilal Subhash. He's a computational postdoc at Simir Bea's lab at Cancer Center of Cold Spring Harbor Laboratory. His primary objective is to explore the molecular mechanisms influencing disease prognosis through the integration of data from diverse high-throughput techniques. Presently, his research focuses on unraveling the complexity of endometrial cancer biology by investigating single-cell datasets obtained from both patients and patient-derived organoids. This involves studying immune cancer crosstalk with the goal of improving treatment modalities for endometrial cancer patients. Santhilal, thank you for joining us today. Go ahead.
Yeah, thanks for the nice introduction. I hope you can hear me. And yeah, so today I will be presenting about one of our unpublished work titled Ancestrally, Diverse Single Cell Atlas of Human Gynecological Carcinosarcoma. So carcinosarcoma is one of the subtype of endometrial cancer, and the recent statistics shows that there is a 1.9% increase in death rate among women due to endometrial cancer, which is a really alarming situation. And also, when we look at the incident and mortality rate, the brown and green line here are the Black women population, which are having increasing rate at 2.4% per year. So this also shows that the incident and mortality rate are more among Black women. And there are different subtypes of endometrial cancer, low and high grade cancer. Today, I will be concentrating on one of the high grade cancer, which is called carcinosarcoma. And it is originated from the inner lining of the uterus. And it has poor prognosis and poor survival. And why carcinosarcoma? So carcinosarcoma is actually very challenging to treat because it is least studied and also constitute 5 to 6% of gynecological malignancies. And this particular tumor is biphasic in nature, which means it has both carcinomatous and such as epithelial-like and sarcomatous such as mesenchymal-like population within the tumor. And also it has 15% of it cancer-related deaths. And when we look at the recurrence rate, it is 50%, and which has for 5-year poor survival rate. Like other endometrial cancer subtypes, from the recent statistics from New England Journal of Medicine, it is shown that carcinosarcoma rates are also higher among Black women. So there are different theories, that hypotheses that have been proposed so far for the biphasic nature of this carcinosarcoma, where this carcinosarcoma tumor undergoes a frequent epithelial to mesenchymal transition. And one such a hypothesis is like collision hypothesis, where epithelial and mesenchymal cells collide each other to form this carcinomatous and sarcomatous component. And the combination hypothesis, where the cancer stem cell giving rise to epithelial and mesenchymal stem-like population, which in turn giving rise to the epithelial and mesenchymal component of carcinosarcoma. So then the conversion hypothesis, where the epithelial cells undergoes transdifferentiation into mesenchymal and epithelial-like states in the carcinosarcoma tumor. So these are the different hypotheses that have been proposed. But it is very important to study this carcinosarcoma in the single cell level to understand the cellular level, like cellular level functionality of this tumor. So for that, we collected a patient sample from ethnically diverse patients and population, and we collected around 13 primary tumors, where one of the patient undergoing metastatic in 2 different stages and 2 different sites, such as ovary and omentum. And we also collected the samples from these metastatic stages. And we also, like when we look at the distribution of this patient, and you can clearly see the higher representation in the African American population, and belonging to the obese category, and also it belongs, they belong to 55 to 75 age group. And this is the whole genome data. So we did admixture-based prediction of the ancestry, and the orange represent African American population. And you can also see that most of the patients are of African American origin. So this is the distribution of the 15 specimens, such as 13 primary tumor and 2 metastatic tumors. Each color represent a patient, and each patient, individual patient. And when we look at the cell types that are enriched in this carcinosarcoma, we can clearly see that epithelial-like and mesenchymal-like components are enriched in this patient, which mimics the carcinosarcoma pathophysiology. And also, along with that, we have stem-like population and cells undergoing epithelial to mesenchymal transition and immune population as well. So when we look at the distribution of these cell components between patients, and you can clearly see that there is a high level of heterogeneity exists between patients. So to further explore these patient tumors, we wanted to segregate the cancer cells from this single cell data. For that, we used a copycat algorithm to predict predict cancer cells and non-cancer cells based on RNA copy number analysis, and we could predict diploid cells that are normal and aneuploid cells that are cancerous. And within aneuploid cells, we also saw multiple clones in each patient. And then this is the distribution of these cellular components, such as cancer, immune, and the stroma component. And this is the RNA expression in each of the component: cancer, stroma, and immune component. And then we also wanted to look at the correlation between the patient in this each component. For that, we did correlation analysis based on top 2000 variable genes in cancer component. You can clearly see that there is less correlation, which means higher level of heterogeneity exists between this patient. And in stroma, we have partial heterogeneity, and in immune, we have like least heterogeneous population compared to other two components. So this is the distribution of the patient in cancer, immune, and stroma components. And you can clearly see that the cancer component has a highly heterogeneous distribution compared to the other immune population. And stroma also has the least, stroma also has some heterogeneity compared to the immune population. So next, we wanted to investigate this cancer component in detail, and we wanted to look at what are the cell types that are represented in this component. So when we look at the cell types that are enriched, we saw most of them are cancer stem cell population, and epithelial-like population, and mesenchymal-like population. And within each population, we have highly heterogeneous cell types represented by different markers. So when we look at the marker expression of this cancer stem cell-like population, we also see high level of heterogeneity at molecular level in each patient represented by different markers. And when we look at the epithelial-like, mesenchymal-like, and EMT population, again, we see high level of molecular heterogeneity exist between the patients. So this shows high level of heterogeneity among patients. So as I said, we also had multiple clones within each patient in this cancer cell population. So we wanted to further investigate these clones, and we did a correlation analysis just based on the RNA copy number. And we can see that these clones, clone 1 and clone 2, are highly similar compared to other clones, other predicted clones. And then we wanted to look at an RNA level. And we did RNA velocity analysis to see the splicing pattern in this clone, to see the clonality in these patients. This is one of the patients where we have clone 1 that is represented by epithelial and stem-like population, and clone 2, which is mesenchymal-like population. And we see that the clone 1 giving rise to stem-like population, and also clone 1 epithelial cells giving rise to mesenchymal-like population. And when we look at the splicing pattern, we can see that epithelial-like genes are expressed early in the pseudotime, and the mesenchymal-like expression expressed in the late in the pseudotime. And we have cancer stem cell-like population that are expressed throughout the stages of pseudotime. So this shows somehow that these epithelial cells undergoing kind of transdifferentiation in this tumor, in this particular patient. We also have other patients that are represented by different cell types, and which are completely heterogeneous compared to this one, where we have epithelial ciliated-like population, cancer stem-like population. We have EMT that is expressed in the later stage. So then we compare our cancer cell population with the available single cell data from normal endometrium for finding the differentially expressed genes. And we found significantly differentially expressed genes, which are mostly associated with downregulated TNF-alpha signaling. And we also have upregulated EMT population. And we also compared our primary tumor with the metastatic tumors. And we could see that in the metastatic, we have opposing pattern of upregulation of TNF-alpha signaling with the upregulation of EMT population. This well corroborates with the either metastatic cancers, so when there is a hypoxia, we see increased TNF-alpha signaling, which in turn maintains the EMT to maintain the metastatic state of the tumor. Next, we wanted to know what kind of transcription factors that are regulating this differentially expressed genes in the carcinosarcoma. So for that, we took the promoter sequence from these differentially expressed genes and looked for the enriched transcription factor over this promoter. And we saw that most of the upregulated genes were enriched with CREB pathways. So this also shows that there is certain level of CREB regulation in these upregulated genes of carcinosarcoma. So next, we investigated the immune population in detail. This is the UMAP representation of the immune population, where we have T lymphocytes, which are CD8 positive, CD4 positive, and T regulatory cells. We also found myeloid cells, which are dendritic cells, monocyte, and tumor-associated macrophages. We also found B cell and plasma B cell population. And when we look at the distribution in individual patients, and we saw higher level of heterogeneity exist in tumor-associated macrophages and regulatory T cells, and also other T cell population, and also the plasma B cell population. So when we look at the distribution of these immune cell population in the normal endometrium, we don't see that much of heterogeneity. We see most of the homogeneous distribution among the donors. And next, we wanted to look at the marker that are present in each patient. And we could again see a molecular level heterogeneity exist in T cell, B cell, and myeloid cell population. Next, we also stratified the patient based on the cytotoxic score, dysfunction score, and Treg activity to know which patient are immune responsive or non-responsive. So we found interesting patient categories that are very responsive and non-responsive patients. And next, we did differential expression analysis compared with the normal endometrium, and we saw that most of the upregulated genes are associated with the T cell toxicity, T cell dysfunction, and also we saw higher upregulation of tumor-associated macrophage compared to the normal endometrium. So as I said, we also have one patient undergoing metastatic stages, and when we look at the signaling that are associated with these primary and metastatic stages, we saw IL-1 signaling that are highly regulated, and when we saw between the stages, we could clearly see there is a higher recruitment of tumor-associated macrophage and other macrophage or myeloid population through when the stage is progressing. And then we looked at the stroma cell population, and in the stroma cell population, we saw higher enrichment of cancer-associated fibroblasts. And in cancer-associated fibroblasts, when we look at the distribution in individual patient, again you see a higher level of heterogeneity in the cancer-associated fibroblasts. And these are some of the markers that are enriched in each of the cancer-associated fibroblasts population. You can clearly see heterogeneous population are present in individual patients. And when we look at the functional significance of this CAF, those are mostly associated with antigen presentation. There is a parasite CAF, which is angiogenesis associated, inflammatory and pro-tumor, and there is also myofibroblast associated fibroblast. So then we analyzed these three components in detail. So we wanted to know how exactly these three components communicating to each other in the tumor microenvironment. For that, we did cell-cell communication analysis between these three components. And when we did cell-cell interaction analysis using ligand receptor interaction, we could clearly see that CD8, there is a higher activity of CD8 positive cells, CAF population. So then we wanted to look at what kind of signaling that are contributing to this higher activity of the cell types. So we looked at the signaling of these cell types. We could see that TGF PVR axis along with CD96, and we saw IL-2 and IL-10, which is mostly T cell dysfunction signaling. And we also see T cell receptor signaling that are highly active. And then we look at the distribution of this signaling in this individual patient. This is the cumulative distribution in the patient, and each color represents different patients. And you can see that PVR TGF CD96 are highly restricted to some patient, not present in other patients. So this also shows the high level of specificity in signaling in individual patients. And so this is the overall study. And with this, I would like to summarize that we found heterogeneity at multiple levels, including patient to patient. also in cellular composition, also within individual cell types. And when we did detailed investigation of individual component, we saw that cancer cells has higher clonality and cellular plasticity. And we also saw that tumor-infiltrated immune cells are associated with T cells toxicity, dysfunction, and myeloid recruitment via tumor-associated macrophages, especially in the metastatic stage. And in the stroma, we found most of them are CAFs, and they are also having a higher patient-to-patient heterogeneity. And we also found TIGIT PVR that are highly specific in particular patients in tumor microenvironment. And it is really like it gives us the opportunity to address the treatment challenges in the carcinosarcoma patients. So with that, I would like to acknowledge our BIOS lab team members and our collaborators at the funding agencies, and especially CGI and the Human Cell Atlas. And thanks for listening. I'm happy to take any questions.
Thank you so much, Santhilal. Are there any questions? We hope to see them in the Q&A section in the last couple minutes. Meanwhile, I would like to thank everyone who joined us today. And stay tuned for the next events in the seminar series. And also would like to thank today's speakers. Thank you, everyone.